The topic for Chapter 7 is cellular respiration. Now, as you recall in Chapter 6, we talked about some of the basic ideas of metabolism, including looking at different kinds of metabolic pathways. If you remember, a pathway is a series of interconnected reactions where we start with a particular substrate, have many enzymes along the way, make small changes to it until we arrive at our final product. Cellular respiration is an important aspect of metabolism in cells. It involves breaking down glucose to extract the energy from it and to put that into the form of ATP, which, as you also hopefully recall from Chapter 6, is the energy currency of the cell, the molecule that's broken down when the cell needs to do work. As a bit of review here, as well as to connect what we're about to talk about with what we've talked about before, take a few moments and think about this question. What is the function of mitochondria? How would you describe their structure? Again, the overall goal here for the cell in respiration is to generate ATP, to make ATP that can be broken down to drive endergonic processes in the cell. There are a few different pathways that are involved here, and we'll go through each one of those in turn. But just as a means of introduction and thinking big picture here, one thing to keep in mind here is that electrons are associated with energy. Within these pathways, you'll see the movement of what are called high energy electrons. Now, if you think about it, you probably already have somewhat of an appreciation for this idea that electrons can have energy associated with them, since what we use to charge our devices like our phones or computers is we plug them into electrical outlets where the flow of electrons then charges them up. We'll see in these processes as well that these electrons can essentially store some of the energy. And again, ultimately, that energy needs to end up in ATP. Now, I mentioned that we're going to be moving electrons around. There is a particular name for the kind of chemical reaction that involves transferring or moving electrons, and that is a redox reaction. So redox actually it's a two-part word. This first part here, the red part, comes from the term reduction. Now, a reduction reaction is one where we add or gain electrons in a particular chemical. The second half of this word here, the ox part, stands for oxidation. And oxidation is the opposite of a reduction. It is a loss of electrons. Now, if you remember, the electron, as I've written here, has a negative charge. So when you have a reduction that occurs and you gain electrons, or the chemical that is reduced tends to become more negative. And when there's an oxidation and a chemical loses electrons, it tends to become more positive. With this transfer of electrons that occurs, electrons don't just go floating off into space or appear from nowhere. Anytime that you have a reduction, you must actually also have an oxidation and vice versa. If there is an oxidation where something loses electrons, another chemical must be there to accept it. So there must be a reduction involved. Because of that, that's why we often combine these two terms into this one term here, redox, because the oxidation and reduction must actually occur together. Because of that, a reaction that occurs where there's a redox often looks something like this. In this case, if you take the A molecule here, notice that it is going from A without a charge to a positive ion. That means that it must have lost electrons. So this is the oxidation. And notice it says here now that A has been oxidized. If you take a look at what happens to the X in this reaction, notice it starts with a positive charge, and then it becomes neutral with a zero charge. That must mean that it gained electrons and hence was reduced. Now notice that in this reaction, the chemical that was reduced is what is called the oxidizing agent. And the chemical that was oxidized is what is called the reducing agent. It's because, again, remember, the oxidation and reduction must occur together. And so even though the X here was actually reduced, in this process, it actually oxidized A and vice versa. A, while it was oxidized, actually reduced X. That's the reason why those labels appear here the way that they are. One way that you can remember oxidation and reduction and the difference between them is this mnemonic. Leo, the lion, says grr, where Leo stands for loss of electrons is oxidation, and grr stands for gain of electrons is reduction. I mentioned that we are going to be moving around electrons in this process of cellular respiration. There are a couple of different molecules that are what are called electron carrier molecules, because they are going to be the ones that will be used to accept and carry these electrons. Now, these electron carriers can have a empty form where it's like an empty bucket that can be filled with electrons, or they have a filled form where they've already been filled up with electrons. Again, we call the empty part where they can still take on electrons the oxidized state, and we call the filled form where they're full of electrons the reduced state. So again, here, let's take this particular electron carrier molecule that's called NAD. NAD plus the oxidized state is what I would call the empty bucket here. And NAD with an H on it is the reduced state. It is what we'll call the full bucket, where this one has electrons and the energy associated with it in it. Now again, the whole idea behind these electron carrier molecules, as we'll see, is that they temporarily hold electrons and the energy that goes along with them on the way to making ATP. Now, there are two electron carrier molecules that we'll see in this chapter in particular. The first one is this molecule NAD here. The second one is another molecule that's called FAD, and we'll see NADP when we look at the process of photosynthesis in the next chapter. Now, just to review a bit about ATP, remember that ATP is the energy currency of the cell. Whenever the cell needs to drive an endergonic process, it will break down ATP. And when ATP is broken down, it forms ADP plus an extra phosphate group here. Another thing that ATP can do is to actually donate a phosphate group to another molecule. So let's say that we have this molecule here, and we spend a little bit of ATP, make it into ADP, take that last phosphate and add it onto this molecule. That reaction might activate this molecule by making it less stable and more likely to react in another chemical reaction. Remember that ATP is a rechargeable molecule. After it's spent to make ADP, if we add another phosphate group back to it, we can regenerate the ATP. And that's actually what this whole cellular respiration process does, is to put ADP and phosphate back together to form ATP. As we'll see in these processes, there's two main ways that this can happen in the cell. The first is to couple this reaction with an exergonic reaction that releases energy. This is what is called substrate level phosphorylation, where there is, say, an enzyme in one of the pathways that catalyzes an exergonic reaction that can just put ATP back together. The second, and actually the way that most of the ATP cellular respiration is generated, is what is called chemiosmosis, which requires a special enzyme that's called ATP synthase. Now, ATP synthase actually exists along the inner membrane of the mitochondria in eukaryotic cells, and hence what we talked about before with the role of the mitochondria in generating ATP. This diagram shows a broad overview of the process of cellular respiration. There are actually three parts to it. The first is what is called glycolysis. The second main one is what is called the citric acid cycle. Now, there is a little bit of a lead-in, a transition, if you will, between glycolysis and the citric acid cycle. That's called the oxidation of pyruvate. And then the third major step of cellular respiration is what is called oxidative phosphorylation. If you take a look here at what's happening, remember that the overall goal of this process for the cell is to make ATP. And notice that in glycolysis, as well as the citric acid cycle, we actually make a small amount of ATP, again, through what is called substrate level phosphorylation. But that's actually not where the majority of the energy goes in these steps. The majority of it is actually funneled via high energy electrons to our electron carriers, NAD and FAD. So again, remember that these molecules are like buckets that we can fill with those high energy electrons, and they get shuttled to this third step, oxidative phosphorylation, where we empty out those electrons and energy and actually make the majority of the ATP in what is called oxidative phosphorylation. Here's the overall chemical reaction that describes cellular respiration. So notice here, this is our sugar glucose. We have to add an input of oxygen into it. And out of it, our products, we get our carbon dioxide as well as water. And again, remember, we're capturing our energy in the form of ATP. Perhaps you've learned in a chemistry class before that when you take an organic molecule, you add oxygen to it and you get carbon dioxide and water out of it. This is actually the general formula for a combustion reaction, which combustion, think something burning, releasing lots of energy. So in essence, the cell is kind of doing the same thing, just in a controlled way and capturing that energy, or at least some of it, in the form of ATP, rather than letting it all just go to heat. So again, there are three main pathways that are involved in this process: glycolysis, the oxidation of pyruvate, and the citric acid cycle, and oxidative phosphorylation. And now we'll talk about these pathways in turn. The first of these pathways is what is called glycolysis. And as we go through here, I did want to mention that there's a lot of information, a lot of details to keep track of, but try to follow along with a couple of main things here. So as we go through, try to think about what is happening to the carbon containing molecules, our organic molecules, and also what is happening in terms of the energy. In particular, for the energy, remember that our overall goal, right, is to make ATP. So at each step here, ask yourself, are we making ATP? Is it a little or a lot? Now, remember, the other place that we see energy coming up in these processes is in filling our electron carrier buckets, right? And remember, these include NAD and FAD. In glycolysis, what we start out with is our glucose that we are going to break down. We also start out with two of our empty electron carrier buckets, NAD+, a little bit of ATP, and some ADP that we're going to make into ATP. Now, glycolysis is a metabolic pathway, which, if you remember, means a series of interconnected reactions, a series of enzymes working together, essentially. And this particular pathway includes 10 different enzymes working together. So they're going to take glucose, they're going to take our NAD plus, and some ATP and ADP. They're all going to catalyze the reactions, and in the end, what we're going to get out of it are two carbon-containing molecules called pyruvate. So that answers that question then of what happens to carbon in this particular pathway. They're going to fill up these two NAD plus buckets to make NADH. They're going to spend a couple of ATP, but in the end, get out of it 4 ATP. This picture here illustrates this point here, that we're going to take our glucose, many enzymes are going to do their thing, and at the end, we're going to end up with these two molecules that are called pyruvate. Now, we can actually split glycolysis down into two parts. The first part is what is called the energy investment phase. So the reason why it's called that is because we actually use up 2 ATP molecules in this process. Remember that we said that ATP could donate a phosphate group to a molecule and activate it by causing it to undergo further reactions? So that actually what happens here in this first part of glycolysis. We spend a couple of ATP and that phosphate group actually gets transferred to glucose or the products that we're making along the way in this pathway. And notice that the end result of this first part of the pathway is to split this glucose molecule now into two pieces. We go from a 6-carbon sugar to these two 3-carbon molecules here. And that's actually where the term glycolysis comes from. The lysis part means to break something apart, if you remember, like hydrolysis. So we're breaking glucose down into two pieces in this part of the pathway. The second half of glycolysis is our payoff phase, where we actually generate the ATP from this pathway. Remember that everything from now on is actually, we're going to do twice because we've made two of these 3-carbon molecules here. So this pathway is going to repeat itself with the second half of glucose that we've made. So again, remember the two questions that I'd like you to consider are what is happening to the carbon? So we've got our 3-carbon molecule here. It undergoes a series of other reactions and ends up making this molecule in the end, again, that is called pyruvate. Now, the second question, what is happening in terms of the energy? Well, remember that we said that we can fill up some of our electron carrier buckets here, NAD, put some electrons and energy into it to make NADH, or a lot of the energy is going right here into this step. We're also making a small amount of ATP in this process, right here and right here. These two enzymes here are doing what is called substrate level phosphorylation. They're actually taking a phosphate group off of the substrate and putting it onto ATP. Remember that we said that glycolysis can actually occur without oxygen available. So some cells that find themselves in low oxygen environments, these are actually the only two ATP that they can produce from breaking down glucose. But we'll get to that. We'll come back to that in a bit. So here's the overall pathway altogether again. So remember that this first half of it is what is called the energy investment phase, where we see glucose being split into these two 3-carbon molecules. And we also see the spending of a little bit of ATP. We actually have to use a little bit of ATP to get this process kick-started. The second half is where we get the payoff, where we actually put energy into our electron carrier, NADH, as well as into making a couple of different ATPs, and we make some more adjustments to our carbon molecule so that it ends up becoming pyruvate in the end. And that's glycolysis. Here's a video that illustrates what we've just talked about with glycolysis. The next main step of breaking down glucose, again, is what is called the citric acid cycle. But before we get there, there's a few reactions that need to take place to prepare us for that. And that is what is called the oxidation of pyruvate. Now, notice that this is actually where this process enters the mitochondria for eukaryotic cells. And again, if we consider our two questions, what is happening to the carbon, first of all, we're taking our pyruvate from the end of glycolysis, chopping a carbon off and turning that into carbon dioxide. We make this 2-carbon acetyl group and attach it to this carrier that's called coenzyme A. So we make, in this process, acetyl-CoA. Now, remember we said the other thing that we need to consider is what is happening to the energy. We don't make any ATP out of this step right here, but if you take a look at what's happening here, we do actually take some of our energy, fill up another NAD electron carrier molecule with high energy electrons. So some of our energy is being shuttled here. Next, we're ready for the actual citric acid cycle. So at the end of the transition reactions, remember we produced acetyl-CoA, and that actually gets fed through this circular pathway that's called the citric acid cycle here. The reason that it's a cycle is because we're going to take this input of acetyl-CoA, or we're actually going to combine it with this molecule here, and then various different rearrangements will be made to the carbon structure as it goes around. creating these various different intermediate products along the way. And at the end, we will regenerate this molecule that will accept in another acetyl-CoA. So that's what makes it the citric acid cycle. It's actually a circular pathway. And when we combine the acetyl-CoA and oxaloacetate, we get this molecule that's called citrate. That's where the citric acid name of this pathway comes from. Now, if you remember, I said to think about what's happening to the carbon here. Along the way, we actually make carbon dioxide here. And in fact, if you remember, what came into the pathway here was this 2-carbon molecule that's attached to the CoA, and we've now lost CO2. So that's actually all the carbon that came from glucose. At this point, it all gets turned into carbon dioxide. Now, for most organisms, carbon dioxide is basically just a waste product. In fact, for humans, we breathe it out, we exhale it and get rid of it that way. So that answers the question then of what happens to the carbon. The second question, remember, is what happens to the energy in this process? Well, just like we saw with glycolysis and the oxidation of pyruvate, we actually fill up more of our electron carrier buckets here. So we've got some enzymes that are going to produce NADH and actually some that produce FADH2. So this is where we see that second electron carrier come in. In addition to that, we also have a little bit of substrate level phosphorylation going on. We directly produce from this pathway 1 GTP or ATP per molecule of acetyl-CoA that comes in here. And if you remember, we actually made 2 pyruvate and hence 2 acetyl-CoA from every glucose. So per glucose molecule, we're actually making 2 ATP out of this cycle. Here's another look at the pathway. So again, highlighting a couple of the important things that's going on here. From the carbon perspective, again, we are releasing the remaining carbon in the form of carbon dioxide here. So after this step, really all of the carbon that came in as part of our glucose is gone. What's happening to the energy? Well, we're making reduced electron carriers. We're making NADH here, and here, and here. And then we're also now using our second electron carrier, FAD, and making FADH2 right there. In addition to that, we are also making a small amount of GTP or ATP. You can kind of think of them interchangeably right there. Now, one of the things that you want to keep track of and consider in this chapter is the numbers of things. So if you think about what we have made so far in this process, we've actually made 4 ATP, 2 from glycolysis, or a net of 2 from glycolysis after we take out what we had to invest to get it started. And we've now made 2 from the citric acid cycle. We've released all of the carbon that came from glucose because remember it was a 6-carbon sugar. We've released it all in the form of carbon dioxide. We've also filled up some of our electron carrier buckets with electrons and energy. We've made 10 NADH and we've made 2 FADH2. At this point, remember the carbon from glucose is completely gone, and so really is all of the energy. It is completely oxidized, and all of the electrons have been removed and shuttled into these electron carriers here. So one question to consider here is, where is most of the energy at this point? Is it in ATP? Is it in the carbon dioxide? Or is it in the electron carrier molecules? Well, if you remember, we said that these electrons that were put into these molecules are high energy electrons. And as you can see here, we've made quite a few of them, right? So most of the energy is actually in the electron carrier molecules at this point. If most of the energy is trapped in our filled electron carriers, how do we get that energy out and convert it into the form of ATP? That's what occurs in the last major pathway within cellular respiration called oxidative phosphorylation. Now, the oxidative part of this comes from the fact that this pathway actually uses oxygen. There are 2 parts to this pathway. an electron transport chain, which is a series of proteins that's embedded in a membrane. In the case of eukaryotic cells, it's actually embedded in the inner membrane of the mitochondria here. A second part of this pathway is ATP synthase, which through the process of chemiosmosis actually generates the ATP. The process of electron transport actually builds up a hydrogen ion concentration gradient, which, if you remember, is one of the types of potential energy. And we actually use the energy from that in order to power the synthesis of ATP. The first part of oxidative phosphorylation is the electron transport chain. And if you remember, as I just said a little while ago, rather than being a bunch of enzymes that are floating in a particular space within the cell, this is actually a physical complex of proteins that are found embedded in a membrane. And again, in the case of eukaryotes, this membrane is the inner membrane of the mitochondria. Now, the proteins are oriented this way, such that part of them touches the mitochondrial matrix. This is the inner space of the mitochondria. And again, where the citric acid cycle is taking place. And then a part of it contacts the intermembrane space. So to the outside here would be the outer membrane of the mitochondria. Again, remember that at this point, most of our energy is actually stuck in our electron carriers, our reduced electron carriers here, NAD and FAD. So what we need to do is to empty them out of both electrons and energy. And so that's one thing that the proteins within this complex do, is they take our reduced electron carriers and then they oxidize them back to their empty oxidized state here. Again, oxidation means a loss of electrons, and our electrons then are accepted by the proteins that exist within this complex. And the reason this is called an electron transport chain is because their electrons are actually passed down the chain here between the different proteins. And as we mentioned, an electron can't just float off into space. There must be something at the end of this chain to finally accept them, where the electrons come to their final resting place. And that's actually where the oxygen comes in. So oxygen in this process of oxidative phosphorylation serves as the final electron acceptor. And when oxygen takes on these electrons, again, since it's gaining electrons, that means it's being reduced in this process. When oxygen is reduced, it combines with some hydrogen ions to form water. Now, there's another critical thing that's going on here. So as these proteins are removing the electrons and energy from our electron carriers, NAD and FAD, they are also pumping hydrogen ions into this intermembrane space here. So they're increasing the concentration of hydrogen ions here versus here. Here's just a little bit more detail about some of the proteins within this complex. So again, the reason that the electrons end up being passed down the chain is because each component is more electronegative and pulls harder on these electrons. So notice that protein complex 1 here is the one that actually oxidizes NADH back to NAD+. It will remove the electrons as well as the H+ from this particular electron carrier molecule. This complex that's labeled 2 here is the one that then oxidizes FADH2 back to FAD. Both of these two pass their electrons to a third component here that's called ubiquinone, which then passes electrons onto complex 3. And then complex 3 passes on its electrons to this portion that's called cytochrome C. Cytochrome C then finally transfers the electrons to this last portion of the complex here, complex 4, where the electrons then are passed on to oxygen, forming water. Now, at the end of this process, remember that not only have we formed water, but we've also created this concentration gradient where there's more H+ on this side of the membrane than this side. Now, remember, when there's a concentration gradient, it means that this ion here that's built up on this side of the membrane really quote unquote wants to come back inside the matrix here. And we can actually harness the movement of that H plus back into the matrix to put together our ATP. You can kind of think about this as how a hydroelectric dam works. Now, if you think about that, you've got a river where water is normally flowing downstream. If you put up a barrier, kind of like the membrane serves here, that will prevent the water from freely flowing, but also create some potential energy because, again, that water really, quote unquote, wants to flow downhill to keep going down the river. And if you put something there in the gateway of that dam, like a wheel that will spin and turn as the water falls, you can capture that potential energy and turn it into kinetic energy. In the same way, this particular protein, ATP synthase, will capture the movement of these H plus ions as they flow back into the matrix and convert that into forming our high energy bond in ATP. The way that the energy from NAD and FAD translates into ATP is as follows. For every NADH, we actually make 2 to 3 ATP. So on average, about 2.5. And for every FADH2, we actually make 1 to 2 ATP. So on average, 1.5 ATP. Now, the reason that this is an average and not a whole nice round integer number here, it does vary based on species and some other conditions. Here's a whole picture of oxidative phosphorylation, including the electron transport chain, where again, we're going to take NADH and FADH2 that have been generated in glycolysis and the citric acid cycle and are holding much of the energy that came out of glucose, empty out the energy and electrons here to form water, as well as to pump hydrogen ions into the intermembrane space. And then in chemiosmosis, ATP synthase captures the motion of these hydrogen ions back into the matrix and uses that energy to put together ATP. Now, returning here to the math behind this, the actual number of ATP that's generated by cellular respiration per molecule of glucose is somewhere in the range of 30 to 32. Now, the efficiency of this actually varies by species, as well as how efficiently NADH enters into the mitochondria from glycolysis. Because if you remember, glycolysis happens out in the cytoplasm, so we actually have to move NADH, transport it into the mitochondria. In thinking kind of more broadly again about the math that's going on here, remember that substrate level phosphorylation, this includes the ATP that we make during glycolysis and the citric acid cycle, is a total of 6. Because remember, we make 4 during glycolysis, but we had to invest 2, so it's actually a net of 2, 4 minus 2. And then per glucose molecule, we actually make 2 in the citric acid cycle. So again, our 6 total comes from the 4 plus 2, and the net of 4 then comes from, if we have 4 minus 2, we make 2 in glycolysis and 2 in the citric acid cycle. Oxidative phosphorylation makes around 28 ATP. Again, if you remember the numbers we said, we make about 2.5 ATP from every NADH. And if you go back and add up the number of NADH we made throughout this process, it's 10. So 2.5 times 10 is 25 ATP that come from this energy that had been temporarily held by NADH. And for FADH2, it's 1.5 ATP per molecule of this, and we made 2 of them during the citric acid cycle. So 1.5 times 2 gives us 3 ATP. And if you add up the 25 and the 3, that's how we get the 28 that come from oxidative phosphorylation. And again, if you compare, remember that this actually makes a net of 4 ATP, and this makes about 28 ATP. You can see really that most of the energy goes this way, temporarily in our electron carrier molecules, and then turned into ATP through oxidative phosphorylation. Now, this might seem like a lot of ATP, but in actuality, it's only about 34% of the energy from glucose that actually makes it into the form of ATP. The rest of it is lost as heat. Glycolysis, if you recall, doesn't actually require oxygen. It can therefore occur in both aerobic, meaning with oxygen, and anaerobic environments, meaning without oxygen. Whereas the citric acid cycle and electron transport are both dependent on that oxygen being able to accept those electrons in the end of the electron transport chain. So, if there's no oxygen available, cells could still do glycolysis and make a little bit of ATP, namely a net of 2 ATP per molecule of glucose. And that's not great, but it's better than nothing, and it might be enough for the cell to survive. There is a little bit of a problem though. If you remember the process of glycolysis, we actually take NAD plus and we reduce it to NADH. And normally in aerobic respiration, during oxidative phosphorylation, remember that the electron transport chain is going to empty out the electrons and energy from NADH. If there's no oxygen though, and hence no electron transport chain, now we've got a bit of a problem. There isn't an endless supply of NAD plus in the cell, and so if glycolysis keeps going without the electron transport, we'll end up using up all of our NADH. It'll all be reduced and turn into this form here. That would shut down glycolysis too, and now we're in real trouble. We can't get any ATP out of this process. But fortunately, in the cell, there is a way to solve this problem, and that is through fermentation. So fermentation is a way of taking our NADH and resetting it, turning it back into NAD+. And that really is the whole goal of this process here for the cell. Now, there are many variations on this, but they kind of fall into two main categories where we create some kind of organic acid, most commonly lactic acid, or some kind of alcohol. Lactic acid fermentation occurs in various different kinds of organisms. It's actually the kind of fermentation that humans do. So when you exercise really vigorously, you exercise really vigorously, so much so that your muscle cells begin to run out of oxygen, they actually begin to form lactic acid. This pathway even exists in some kind of microorganisms. And in fact, the bacteria that are those that make yogurt out of milk do lactic acid fermentation. It's actually the lactic acid that gives yogurt its distinctive tart flavor. Now, this is a pathway like the other pathways we have seen, so there are enzymes that catalyze the reactions here. Notice that both kinds of fermentation, including lactic acid fermentation, we're actually going to start with our product from glycolysis pyruvate and our filled reduced electron carrier bucket NAD. And out of it, we're going to make lactate and reset NAD+. Again, remember, this is the whole goal overall of the pathway here. The other kind of fermentation is what is known as alcoholic fermentation. Now, the organism perhaps best known for doing this type of fermentation is yeast. And in fact, it is the action of yeast that produces various different kinds of alcoholic beverages. Now, notice that similarly to lactic acid fermentation, we start with pyruvate, which again is the end product of glycolysis. From it, following a series of reactions, we make ethanol. Now, one thing that occurs in alcoholic fermentation is actually the production of carbon dioxide, CO2 here. But again, remember, the whole purpose of this reaction is to take our filled electron carrier molecule, NADH, and to convert it back into the oxidized form, NAD+, again, so that we can keep glycolysis going and make that little bit of ATP. Now note that with both alcoholic fermentation and lactic acid fermentation, we actually make no ATP. We make 0 ATP directly by this process. But the reason why these reactions are advantageous to the cell, again, is to reset our NAD+ so that we can make ATP from glycolysis. So far, we've been talking about glucose catabolism, breaking down glucose, kind of in a vacuum. But you have to remember that metabolism includes all of the chemical reactions that are happening in the cell. And these pathways that we've talked about are in fact connected to other pathways that occur in cells. For example, these pathways can be used to break down some other kinds of molecules in order to get energy in the form of ATP from these other molecules. For example, some other kinds of carbohydrates, other similar sugars, can be modified slightly by enzymes so that the products from them can then go through glycolysis. The same is true with some amino acids and glycerol, which again is the backbone of our fatty acid containing lipids. The fatty acids themselves can be broken down into small pieces through a process that's called beta oxidation, and then those pieces can be fed through the citric acid cycle. The different kinds of amino acids, if you remember, all have those different R groups, those different structures, and hence different enzymes can modify them to make molecules then that can enter into these different steps within respiration. One thing to note about this, about the interconnectedness of these pathways, is the citric acid cycle is really a major hub of metabolism. We mentioned that some of the intermediate products that are made in the citric acid cycle can be shuttled away to make certain amino acids, and the reverse is also true. If the cell, say, has some old proteins that are being degraded and actually has a need for more ATP, we can feed these amino acids. We can have some enzymes make some modifications to these amino acids to make them into pieces that can go then through the citric acid cycle here. And these different types of amino acids will enter into the citric acid cycle at different places. So for example, the amino acid glutamate through this enzymatic pathway here can be converted into this intermediate alpha ketoglutarate and enter the citric acid cycle. But if you have an asparagine amino acid instead, this different pathway will convert it to oxaloacetate and it'll enter into the cycle here. Now, these pathways are actually regulated by different mechanisms, and there's actually different levels of control here that can occur. The first level of control is whether or not glucose actually enters into a cell and is taken up by a cell. There is a peptide called insulin, and insulin is released or secreted into the blood when there's a high level of glucose that then needs to be taken into cells and broken down. So you can see here, insulin binds to a receptor. We'll talk in future chapters about cell signaling, but this is one example of that. When insulin binds to this receptor, it creates changes inside of the cell that bring this protein channel to the membrane and then allows glucose to enter into the cell. If you recall, during our last chapter, we talked about some of the feedback mechanisms that can control a pathway, and we see that actually occurring in the pathways of cellular respiration. So for all of these pathways, there are certain key enzymes that are regulated by these effector molecules. Some of them, like this one here, glucose 6-phosphate, is a product of the reaction that's catalyzed by a hexokinase here. And so if there's a lot of this product already, then this enzyme doesn't need to be as active. So this can actually serve as an inhibitor of the enzyme and prevent there from being too much of this product accumulating. Same is true of this one. We have the enzyme pyruvate dehydrogenase that's involved in making acetyl-CoA. Well, if we already have a lot of acetyl-CoA, then we can use that to inhibit this enzyme and slow this process down. One thing to note in general is that ATP often serves as an inhibitor and decreases the activity of many of these pathways. If we have a lot of ATP, it's less critical to make more at the moment.